Good morning, children. How are you? I hope you all are fine and you have gone through the topic we discussed in the previous video. We were discussing about the conditions which are necessary for photosynthesis. In our previous video, we discussed that how chlorophyll is necessary for photosynthesis. We did an experiment where no chlorophyll was present and there was no starch being formed in the leaves. We discussed about how sunlight is necessary for photosynthesis. We discussed one experiment. Now the third condition is that we all know carbon dioxide is also necessary for the process of photosynthesis. So to prove it, we will do one more experiment. In this experiment, what you need to do is take two potted plants exactly similar and same in size also. Now, we need to keep these two potted plants in a dark place for 2 to 3 days to destarch their leaves as we had been doing in the previous experiments. When we will keep it in the dark place, there will be no sunlight, no photosynthesis will be will occur in the plants, and moreover the starch which is already present in the leaves will be utilized by the plants. Label them, mark them as plant A and plant B. You can mark or paste on the pots itself. Now place each plant on a separate glass plate. Better, this is the separate glass plate. We have put these plants on the glass plate, and in the setup A, what we have done is we have taken a watch glass which contains potassium hydroxide solution. But in the setup B, we need not to put any watch glass as such. After that, what you need to do is we need to put two bell jars over each plant. After putting the bell jar, we need to seal the jar with the glass plate by applying Vaseline over this. Now what will happen? In the setup A, there is potassium hydroxide, so the air or the carbon dioxide which is present in the bell jar will be absorbed by the potassium hydroxide solution. But in setup B, there is no potassium hydroxide solution, means there is carbon dioxide within the bell jar. Now, after doing this, we need to keep the plants in sunlight for 3 to 4 days for normal photosynthesis to happen. After 3 to 4 days, pluck a leaf from each plant and remove the green colored chlorophyll from the leaves by boiling it in alcohol as we did in the previous experiments, because chlorophyll interferes with the test of the starch. Now after that, what you need to do is pour iodine solution over both the leaves. What you will observe that in plant A, the leaf, when you will put iodine, it will not turn blue black, but the leaf plucked from plant B will turn blue black. This means that in plant A, no photosynthesis has occurred, but in plant B, photosynthesis has taken place. Why is it so? In setup A, there is a watch glass which is containing potassium hydroxide, and this potassium hydroxide absorbs entire CO2. So when there is no carbon dioxide, no photosynthesis will occur, no starch will be formed. In setup B, there is no watch glass. So in the jar, there was already CO2 present. So that CO2 helped the plant to carry out photosynthesis, and little bit starch was formed which gave the color with the iodine solution. So with this, so far we discussed that sunlight, chlorophyll and carbon dioxide all are necessary for photosynthesis. In the absence of any one of these, photosynthesis cannot occur. But the other important raw material which is required for the photosynthesis is the water. Remember beta, water is taken from the soil by the roots. Roots absorb the water by the process of osmosis. Other than this, other nutrients like nitrogen, phosphorus, iron, magnesium are also taken from the soil. Till now, we discussed about all the conditions. If you remember my previous video, we discussed how the plants obtain water and carbon dioxide. I am giving a review of that also. We all know that there are certain tiny pores called stomata on the undersurface of the leaves. In the broader leaves, they are present on the undersurface, but in the narrow leaves, they are equally distributed on the upper and the under surfaces. So, carbon dioxide enters through these stomata through a specific procedure. Opening and closing of stomata is controlled by kidney-shaped cells called as guard cells. If you see the structure, it is a stomatal pore, and these are the bean-shaped cells which we call as guard cells. What happens is, from the epidermal cells, when water enters into the guard cells, these guard cells, they swell up. When these guard cells swell up, they become turgid, they become curved and cause the stomatal pore to open. Same way, when these guard cells lose water, they shrink, they become straight and cause the stomatal pore to close. You could see, and after writing these 2 points, you need to draw diagrams also. So, bachche, you need to remember certain points that when plants do not need carbon dioxide, at that time stomatal pores remain closed, because we know that there is a loss of water, a lot of water gets lost from the stomata. So to conserve water, these stomatal pores remain closed when plants do not need CO2. Oxygen gas produced during the process of photosynthesis is also released out through the stomata. So in the nutshell, we can say that exchange of gases in the plants occurs through stomata. This is all about nutrition in plants. Up till now, beta, we have done all these questions. I have given you the questions also, and all these questions must be written in the notebook. There are 17 questions, and in our doubt session for Saturday, I'll be discussing, I'll be making you mark these answers in your Lakmeer Singh book. So bring the book along with you and do attend that doubt session so that you are able to get the answers also. You are clear up what is to be written along with this question. Thank you.